Hello, medical students. Today, we will be discussing an important technique in molecular biology called polymerase chain reaction, PCR. PCR is used to amplify specific DNA sequences, enabling their analysis and identification. Throughout this lesson, we will explore 12 different variants of PCR, each with its own unique mechanisms and applications. Let's get started with our first variant, nested PCR. Nested PCR is a technique that enhances the accuracy of PCR amplification. It involves 2 rounds of PCR using different sets of primers that target the same DNA region. This approach eliminates nonspecific amplification, making it valuable in complex samples with low target DNA. Hot start PCR is a modified technique that reduces errors in DNA amplification. It involves adding an antibody or chemical to prevent the DNA polymerase from working until the ideal temperature is reached. This improves the accuracy and efficiency of the PCR process. Allele-specific PCR is a technique that detects genetic variations at the nucleotide level. It uses specific primers to selectively amplify either the wild type or mutant allele. This method is useful in genetic diagnostics and understanding genetic disorders. Methylation-specific PCR, MSPCR, is a technique employed to detect DNA methylation patterns, which play significant roles in gene regulation and epigenetic modifications. This variant utilizes bisulfite treatment of DNA samples to convert unmethylated cytosines into uracils, while methylated cytosines remain unchanged. Following bisulfite treatment, MSPCR employs specific primers designed to amplify either the methylated or unmethylated DNA sequences selectively. Differential amplification using these primers enables the detection of DNA methylation patterns associated with gene expression, imprinting, and cancer development. Inverse PCR is a method to amplify DNA sequences next to a known region. It involves cutting and joining DNA to form circular templates, which are then used in PCR. This technique helps study gene rearrangements and identify target sequences. MLPA is a technique used to identify and measure changes in the number of copies of genes and rearrangements in the genome. It achieves this by combining the ligation of multiple DNA probes with PCR amplification. MLPA is valuable for diagnosing genetic conditions and identifying disease-related genetic changes. Digital PCR, dPCR, is a breakthrough method for precisely measuring DNA molecules. It divides the DNA sample into many reaction volumes, amplifies them, and then determines if the target DNA is present. This technique is highly sensitive and accurate, enabling applications such as liquid biopsies and disease detection. Rolling circle amplification, RCA, is a powerful isothermal amplification technique used to amplify circular DNA templates. This method utilizes a DNA or RNA polymerase, along with a single primer that hybridizes to the circular template DNA. The primer extension by the polymerase enzyme results in the formation of concatenameric DNA copies, which can reach thousands of nucleotides in length. RCA has proven highly valuable in applications such as whole genome amplification, circular DNA detection, and the production of nucleic acid probes for in situ hybridization. Cold PCR is a modified PCR technique that enhances the detection of low-frequency mutations or rare genetic variants present in a heterogeneous DNA population. This variant exploits the differential melting temperatures, TM, of wild-type and mutant DNA sequences. During the denaturation step, cold PCR utilizes lower denaturation temperatures than conventional PCR. This selective denaturation allows preferential amplification of mutant DNA sequences possessing lower TM values. Cold PCR greatly improves the sensitivity and specificity of mutation detection, making it ideal for diagnostic applications and cancer research. Preamplification PCR is a technique utilized to amplify limited or precious DNA samples, such as those obtained from small biopsies or single cells. This method involves a preliminary PCR amplification step, utilizing universal primers targeting multiple regions of interest. The preamplified DNA product is then diluted and subjected to subsequent amplifications targeting specific DNA sequences of interest. Preamplification PCR enables the production of sufficient DNA quantity for downstream applications, while maintaining compatibility with limited starting material. Bridge PCR is a variant of PCR mainly used for the amplification of short DNA fragments, including those obtained from next-generation sequencing, NGS, library preparation. This technique involves the ligation of specific adapters to both ends of the DNA templates. During PCR, one primer hybridizes to the adapter sequence, while the other primer anneals within the DNA fragment. This bridge structure enables the efficient amplification of short DNA fragments without introducing nonspecific amplification. Bridge PCR is widely employed in NGS library amplification and molecular diagnostic applications. Anchored PCR is a technique that allows scientists to amplify and study DNA regions next to a known sequence. It involves attaching a known DNA sequence to the target DNA and using specific primers for amplification. This method helps identify new genes or gene fusion events. PCR is a vital tool in molecular biology, with diverse and versatile variants that cater to research needs. Mastering these advanced techniques will benefit your medical career in understanding genetic diseases and facilitating personalized medicine.